Our presenter today is Professor Ashley Shade. Dr. Shade received her Ph.D. from the University of Wisconsin in its microbiology doctoral training program in 2010. She was subsequently a Gordon and Betty Moore Foundation postdoctoral fellow in the Life Sciences Research Foundation at Yale University. In 2014, she started her position in the Department of Microbiology and Molecular Genetics at Michigan State, joining the Department of Plant, Soil, and Microbial Sciences with a partial appointment in 2017. She is part of the Plant Resilience Institute, a research program funded through the Global Impact Initiative, and serves as a project lead with the Great Lakes Bioenergy Research Center, funded by the Department of Energy. Dr. Shade serves as an editor for the American Society of Microbiology Journal, M-Systems, and for the journal Ecology Letters. She is an early career fellow of the Ecological Society of America and the recipient of a National Science Foundation Early Career Award. Her research program is funded by the U.S. National Science Foundation, the U.S. Department of Agriculture, and the U.S. Department of Energy. As a special note, she's just returning from a special interagency workshop on deciphering the microbiome, which was held at the National Science Foundation in Washington. There's every prospect that the National Science Foundation, the National Institutes of Health, the Department of Energy, and USDA, which were all present at this interagency workshop, will fund microbiome research in a significant way in the near future. Let's welcome Ashley.
Good morning. Thank you so much for the opportunity to share a little bit about my research program with you today. But before I get started in the science, I wanted to share with you something special about Michigan State that really drew me to accept a faculty position here when I was on the job market. So you may not know, but Michigan State is home to the original Center for Microbial Ecology. This center was started in 1976 by National Academy member Jim Teege, and it was the inaugural Science and Technology Center funded by the National Science Foundation, and then it was funded for an additional 10 years by the state of Michigan. And what this center did for Michigan State is that it put Michigan State on the map as the place to be conducting research in environmental microbiology and microbial ecology, and it maintained that reputation for about 30 years at the forefront of the science. And it is because of that reputation and because of the legacy left by this center that now we have numerous new microbiome centers all over the nation and also abroad, following in the example of Michigan State. And so this is a big push, especially in the past 3 years. We've had a lot of new microbiome centers opening because of the interest of the federal funding agencies in continuing this very important line of research. So about my research program, we are focused on microbiome resilience. And today I'm going to tell you a little bit about the middle program, which is microbial controls in agroecosystems. But you may have heard this term. What is a microbiome? You've probably heard of it in regards to human health. It's in the news a lot, right? But a microbiome is simply a community of microbial populations that live together and interact with each other with their local environment. And when scientists talk about the microbiome, they're usually focused on understanding that community as a system, including the genes and the molecules that create those interactions and make that community function healthfully. So humans have a microbiome, lots of other things have a microbiome too, including plants, and that is part of my research program. And so, like the human microbiome provides important contributions to our human health, plant microbiomes also provide very important contributions to plant health. In particular, they help plants access nutrients, they help to suppress pathogens, and they also help plants to tolerate stressors in their environment, like drought. Something that you should know about the plant microbiome is that it is incredibly diverse. A single gram of soil can contain tens of thousands of different microbial populations, and a root microbiome can contain thousands of populations. These communities are also very dynamic. They change over time and in response to plant signals over plant development and with a changing season. And so when we study the plant microbiome, we're very concerned with trying to figure out who all those members are and what they're doing to benefit the plant. When we study microbiomes, we study them as a system, and we use some of these really new technologies to try to observe very precisely the microbiomes themselves and also the chemical environment that they inhabit. And to do this, we need some special resources, special technologies. My lab uses mass spectrometry to precisely characterize the physical and chemical environment that the microbes are making and responding to. We use genomics to characterize the genes that those microbes provide to underpin the functions that they are doing. And we use a lot of computational resources to do the heavy lifting with the analysis. So at Michigan State, we take use of the mass spectrometry core, the genomics core, and the Institute for Cyber-Enabled Research to support this program. So I told you what a microbiome is, but I also wanted to tell you about resilience. Resilience is the capacity of a system to recover after it's been altered by a disturbance. And I am passionate about understanding microbiome resilience because it is urgent and important given ongoing stressors of our planet to global climate changes. So what I'm showing you on this slide is a figure that was recently published, 2019, by a consensus of microbiome researchers. And the objective of the consensus report was to try to link the processes that microbiomes provide in the environment and with plants and other systems to global climate change feedbacks. And so there are lots of very intimate relationships between microbiome functions and our changing climate. And so what my research program's pie in the sky goal is to try to understand and manage microbiomes towards stable functions despite a changing environment. And that means we need a really intimate idea and quantitative understanding of how they're responding to their environment and what makes them stable. So I told you that I was going to be working on plant systems. So part of my research program is in the plant microbiome. And luckily for me, Michigan State has excellence in plant and agricultural research. We have a great reputation here. My plant microbiome research is conducted with the Great Lakes Bioenergy Research Center and also with the Plant Resilience Institute. I'm going to tell you just a little bit of what I've got going on with the Plant Resilience Institute. So the Plant Resilience Institute is part of the Global Impact Initiative. It was started about 3 years ago, and it brings together different scientists with different interdisciplinary perspectives for collaborative research to try to understand how plants respond to changing environments and stress in their environment so that we can accomplish goals to maximize food security. So we've got a lot of different projects going on in the PRI, but one pilot project that we started as a team was to try to understand the stress tolerance of common bean. You may be like, well, why common bean? Well, common bean is a really important food and nutrient source in developing nations, so we're interested in helping to provide food security in those nations where global climate change is expected to decrease production. But also common bean is really important for the agricultural economy in the state of Michigan. We are the #2 exporter of common bean in the United States. We grow a lot of black beans in the state of Michigan. And so we wanted to give back to the state and try to understand common bean given global climate change. And so with my group, we started a bunch of different microbiome projects, all related to understanding the microbiome and the beneficial members that associate with the common bean. And I'm going to show you just a little bit of data from one of our first experiments that were quite exciting. And so this is a project where we were looking at the microbiome that is packaged inside the seed of the common bean. So that's the part that we eat. They make a very big seed. That's why they're so delicious. And so what we did is we took bean plants and we grew them in the environmental growth chamber. So this gives very controlled environmental conditions. We're controlling the temperature, the day-night cycle, everything. And we subjected some of those plants to stress, drought stress, and also excess fertilizer. And then we grew some plants under normal, optimal conditions. And when those plants set seed, we investigated the microbiome that was packaged inside the seed. And what we found was that the plants were stressed had a fundamentally different diversity and composition of their microbiome than the plants that were not stressed. And this is a really important observation because that packaged microbiome is what the parental plant is passing on to its offspring. It's essentially the microbiome that the plant starts off with in the next generation. And it can have important implications on the health and well-being of that plant for the next crop cycle. And so what our lab is doing now is following up on these results, is trying to understand exactly how that microbiome is altered, which members are particularly enriched and which ones are depleted, and whether those particular memberships can help the next generation of plants withstand the same or different stressors, or if they have consequences for the growth or productivity of the plants. And so this is ongoing work. We're really excited about it, and I have 2 great graduate students helping me with this project. Before I sign off, I just wanted to give you the face of the next generation of microbial ecology and evolution at Michigan State. We're all on the tenure track, and we're all really excited to be carrying the banner forward of Michigan State's excellence in microbial ecology. And I'm very happy to take any questions you may have. Thank you for your time.
Questions? I'll just ask one very quickly. What is how do you test whether or not a given class of bacteria or group of bacteria may be actually contributing to this response? And so what are the ways, because you have this great huge mix, so how do you take a look at a specific segment?
That's a very good question. So the question is, how do we figure out which microbes are important and go about testing causality in a response? And so I mentioned that the microbiome is often studied as a system. That means that the expectation is that there's not just going to be one member that's responsible for any benefit or detriment to the next generation of plant. That being said, we've generated an isolate collection from the seeds that were stressed, and we're hoping to put them back in different combinations and in a controlled manner give them to the plant so that we can test our hypothesis. There's another way that we go about this, which is statistically by looking at how the microbiome changes over plant development and with different plant compartments. We can statistically figure out what we call the core membership. So these are members that are uniquely associated with the plant consistently, and we target those members when we're going back and trying to isolate for our experimentation.
Great, thank you. So again, excellent work, and thank you so much.